AP Biology. Hope you're doing well when you see this video. So I'm trying to make a video. It's been a long time since I made these, so you'll have to excuse me if I'm a little rusty, but the purpose of this video is to go through the protein notes at home so you can actually have me talking through them and seeing what's important, but also for me to kind of share with you how I would study as I go through. I think a lot of you are kind of in this position of I want to do well, not sure how to study, not sure what I should know, and unfortunately the study guides aren't helping, which I'm going to try to go through some of the study guides as well at the end of this video for the test coming up next week. So this is my effort to try to help you. Hopefully it does, Lord willing, that's my prayer. So let's go ahead and get started. So we're going to go into proteins. Okay, so we've covered the macromolecules of life, the biomolecules, carbohydrates, lipids, nucleic acids, and proteins. And so last one's proteins. Okay, so proteins are the most diverse group of class of biomolecules, okay? And so you can see right here, this, for example, proteins help with metabolism. So all of these enzymes right here, you can see they end in ASE. I would know that for sure. Sorry, I'm writing with a mouse. ASE, but all these reactions happen with an enzyme, you know? Proteases are enzymes that act on proteins, lipases. And so, for example, on this slide, would defining all these enzymes be important? No, that would not be something that we would memorize. But we need to know what enzymes do. And enzymes help with metabolism, and which are catalytic molecules that speed up reactions. Catalytic means they kind of lower the activation energy of reactions. Okay, defense molecules. Sorry, I'm just trying to take away this. Okay, functions of proteins. Defense. Yeah. So this is an antibody. We know all too well about antibodies as far as COVID lands go. And so we respond to substances and target them for destruction for our immune system. So proteins do that. Proteins also regulate and physiological processes, and also they involve reception, which means receiving and responding to molecular signals. So, for example, a great example of this is glucose in the blood. When glucose is in the blood, there is receptors that notice that, like, oh man, we have a spike in glucose, and so then we start making something called insulin. And insulin binds to a protein that allows the body cells to take in glucose. And so that would be important. And now it's a negative feedback example. Functions of proteins, for example, there's storage and structure and support. An example of this would be collagen. So this is what you should know, right? This is a function of the protein. An example of this is just to help you understand that function, okay? So lip injections is type of collagen type of injection. So there's a famous, I guess this is a Jenner, I'm not really sure, I don't remember. I was going to ask y'all actually, but yeah, there's collagen injections, and of course like AI fixing a photo and stuff. But nonetheless, functions of proteins, yeah, they involve transport. So hemoglobin is an example of a protein that transports, and it transports oxygen around. Of course, crazy important for us to have oxygen around. And one of the harder things that we're going to go over is regulation this year, and how genes are switched on and off, and homeostasis and all that. But they're regulatory proteins, which means here's like this really complicated picture that you don't need to know a lot about right now. What you do need to know is that all of these shapes are proteins. Okay, for our purposes right now, and these shapes determine if this gene is being transcribed, right? So remember, it's DNA to RNA to protein, right? But we're not making all the genes all the time. That would be very inefficient and also cataclysmically destructive. And so how do we know when we need a gene to be read and transcribed into RNA and then made into a product or a protein? Well, proteins are around. The environment determines that. Which proteins are around determine kind of that feedback mechanism. Movement, yeah, proteins are involved with movement. Actin and myosin are two examples of this in your muscles. And these are attracted to each other, and the sliding filament theory allows your muscles to move. That would be important about that. So there's 20 different amino acids. Of course, you do not need to memorize all of them. But you do need to know that this is an amino acid. You have to know this. I would make a note card. I'm all about note cards. Making note cards. If you haven't made note cards, just for the record, I will tell you to do that first and do them really well, and then see how you do on tests. But this is a note card. On the front of the note card, hey, draw an amino acid. And then on the back, you have your answer, but you try to do it every single time you see it. And so an amino group is right here, just the functional group. And then you have the carboxyl group, which is on the opposite side. Then you have your carbon, have a hydrogen, but the side chain is where everything gets its properties. I'm just going to write properties. And so this side chain changes, try saying that fast, depending upon what molecules are inside of it. And so how is diversity of proteins created? Well, the diversity is created because whereas there's 20 different amino acids, combinations of 2 amino acids can mathematically generate 400 distinct dipeptides. And so you can see that even a small peptide of 100 amino acids has this many possible sequences. So you can see literally the possibilities are limitless. What a complex creation we're learning about, right? Points to God. So you can see amino acids. You do not have to memorize this. You do not need to know this. But you do need to know, if I show you pictures of them, you do need to be able to see, and let me see if I can highlight instead of draw. Let me see if I can do that. Maybe not. Maybe not. Yeah, highlight, sweet. You do need to see these positives. Polar. Negatives. Polar. Right? Hydrophilic. Going to interact with water. That's important. Okay? Amino acids with polar but uncharged sides. So these are polar molecules still, but this is because of unequal sharing of electrons, possibly. Benzenes, of course. Special cases. Glycine, proline. Hydrophobic. That's a key word to kind of see right there. So these would not want to interact with water. These are hydrophobic. Notice all the carbons and hydrogens. Carbons and hydrogens as well. And so basically how a protein looks determines what it does. And so depending upon the different side chains, you have different side chains available. And how that protein is made based upon the side chains determines what shape it is. And that shape determines its functions, which I'll tell you about more in a second. So polypeptides are just larger polymers. Remember that keyword polymers. We're building polymers from monomers. And so polypeptides means many different peptides. And peptides are joined, amino acids are joined in peptide bonds. So it looks like this. See, water is leaving. So what type of reaction is this? I'll let you hopefully answer that correctly. Hopefully you said dehydration synthesis. And so we're building, right? There's the peptide bond right there. Super important to see that. And so protein structure, this is a very big learning objective. You need to know the 4 levels of protein structure. So these are note cards waiting to happen. What's the first level of protein structure? What's the second level? Third level? Fourth level? These are note cards waiting to happen. So the precise sequence of amino acids is the primary structure. So for example, here is the precise sequence of the hemoglobin beta subunit. All these letters correspond to a specific amino acid. You can see there's roughly 140, 50 or so. So this is the primary sequence. It is, okay, this amino acid followed by this amino acid followed by this amino acid. That's the primary sequence. The secondary sequence consists of repeated spatial patterns. Let's stop highlighting and let's do freehand. Repeated spatial patterns. So for example, we're talking about alpha helices, or the alpha helix. So it's kind of like spirals, right? Different ways it can spiral. Coiling comes from where? Hydrogen bonds. Okay? Or beta sheets. Again, happening because of hydrogen bonds. Okay, that's all you need to know about that. Tertiary structure is when these R side chains start interacting. And so they form disulfide bridges, which is this guy. Hydrogen bonds, of course, always. Hydrophobic side chains want to stick together inside the protein because it doesn't want to be near water. Van der Waals forces, which is unequal sharing of electrons, creates small negatives and positives in a molecule. And ionic bonds, which is donating electrons. So this all involves a tertiary structure. Great note card to make. And then finally, you have, and this is kind of what it looks like, ionic reactions. There's a donation of electrons there. This is what a protein looks like from another book. So this is computer generated. You can see the alpha helices. These are beta sheets. And then you would expect hydrophobic groups inside, hydrophilic groups on the outside, exposed to water. And so then the quaternary is sometimes, it doesn't always exist. Sometimes the protein is completely ready to work in its tertiary form. But some functioning proteins need like many different subunits bound together. So when you add threes together, so this is a third structure, this is the tertiary, these are all tertiary structures, and they combine to form a functional protein, and that's like hemoglobin. Okay, proteins can become denatured. I don't know if you like eggs or if you cook eggs often, but frying an egg is an example of denaturing. So an active protein has a certain shape. It's all about shape leads to function, right? It has a certain shape, and that shape is denatured, and the protein no longer works. What can denature a protein? Have to know this. This will actually be a lab down the road, as we investigate it. But you have to know these things. Temperature can change a protein shape and denature it, which means it no longer works. pH, high concentrations of polar substances. Why? Because it disrupts the bonding. Non-polar substances. Why? Because it can mess up with the bonding. All of this will change. It's super important, right? Shape. Shape determines function. Shape. All of that will change the shape. Okay? Protein folding diseases. This is from another textbook. I would generally know this. This isn't like a huge learning objective, but yeah, there's a lot of diseases that are involved when a protein is not formed correctly. Chaperone proteins help proteins fall into their normal shape. So defects in chaperone proteins, this is just an example, may play a role in like Alzheimer's, so even cystic fibrosis. Prions are misfolded proteins that may have been implicated in failed brain diseases. Mad cow disease is an example of that. Okay, and so proteins interact with other molecules. Whenever the shape changes, we possibly, not 100%, but possibly could change the function. You can see right here, proteins can change shape. And so here's an example of an unbound molecule, and then we bind it. So enzymes do that, right? There's an active site. Sometimes we can modify an amino acid and turn on a protein, activate it per se. And so we can do that as well. Protein, and that's just kind of reviewing. And so here's the questions that you can work on. We'll go through some of these on Monday, Lord willing. What are two common secondary structures? How are they stabilized? Hair, think about how hair works. Okay, now let me go now to, if I can, the study guide. Okay, so this is the study guide I sent out for chapter 2. And so I'm a firm component of note cards, right? And explaining stuff out loud. These study guides should help. But remember, you have to know the concepts so well that you know the concepts and you apply it to something brand new, like a new experiment, a new type of data that you've never seen before. But those questions are important because we have to be able to critically think. So it's not just regurgitation. Yes, there's going to be memorization answers on the test. Yes, there's going to be just basic remembering, basic explaining, basic describing. Yes. But you also have to be able to take the knowledge that you have and apply it to a new situation, a novel situation, and show me that you know what you're talking about. Okay? And so these are the proteins there. Again, I see so many note cards waiting to be made here. Use action verbs, right, on your note cards. Don't just write high heat capacity, explain. Like you start practicing these action verbs. Explain high heat capacity. Do various things like that. This is the chapter 3 one. Contrast hydrophobic and hydrophilic. Understand the 4 macromolecules. I mean, this is like one bullet point, right? And so I don't want to take up your time going through all this because I know you know, of course, how to read, and y'all are all so smart. I'm so proud of all of you already. We're gonna have a good year, but yeah, it's a hard class, and I want to do my best for you. But understand the 4 macromolecules, form and function of each, and know each one's monomer. That's one bullet point, but I could literally probably make like 20 note cards out of that, if not more. Okay, and so yeah, drawing it out, understanding it, like that. That's a huge bullet point right there. Okay, how do functional groups affect a carbon's molecule chemical reactivity? Well, you gotta start thinking about polarity and all that sort of stuff. Relate monomers and polymers. Well, that relates to that right up there. Summarize the role of carbohydrates. That is also that basically. And I just kind of go through the different ones with you. Carbohydrates and lipids. Triglycerides, what are they? Fats and phospholipids and steroids and waxes. Differences between DNA and RNA. And then Here's compare and describe. I mean, that's not one note card. That's like many, many different note cards. You're like, man, this sounds like a lot. Well, that's why we study gradually. That's why we don't wait until last minute. Like this weekend, I strongly suggest you going through and making note cards. So that way Monday night, you have all the note cards made and you're just reviewing Monday night. Okay? So, in conclusion, I'm really proud of you. Like, know that from my heart. Like, I understand y'all are taking really hard classes. I understand you have really demanding schedules. I understand all of that. I understand that we come from lands, in education where it's basically just regurgitation, like memorization type questions. Like, hey, I need to know primary structure, and he's going to ask me on the test, what is the primary structure of a protein? Define it, right? And that's good. Like, that's the foundation of knowledge, but that's not where we stay in this class. You have to take your knowledge and apply it to new things, apply it to new questions. So for me to go through and exactly tell you every single question on the exam isn't prepping you for May and isn't prepping you for college. Me going through the concepts with you and helping you become critical thinkers and teaching you how these concepts relate to each other, Lord willing, and then you being able to know the concepts so well that you apply it to something new, that is where this all kind of comes into play. But I am trying to give you an olive branch here. If you go back to the test, I gave you all the essay questions. You can see this right here. And wow, there's like a little space there. Wow, I'm gonna resend that out because that does not look good. I'll resend that out. But I would go through and answer all these questions. I'm not going to have all the questions on the exam, but you going through and answering all of them, not using any sort of ChatGPT help. But yes, looking up answers, that's fine. Researching answers, that's fine. But writing out the answers and explaining it to yourself. Do you understand what you're writing, or are you just writing down to get a grade? There's a big difference. Am I understanding like this concept? Do I understand the importance of it, how it relates to other concepts, or do I know the answer? And if the answer is not exactly worded on the quiz, I'm going to get it wrong. That's a problem, right? You have to know why the answer is important, how it relates to other things, not just know the answer to get the answer right on a test. There's a big difference in that. I think y'all probably understand what I'm talking about, hopefully. And so we'll try our best to review a little bit on Monday, since you just took the last notes on through this. And yeah, I'm looking forward to it. I hope y'all have a blessed weekend. Yes, study. Yes, start doing some note cards. Remember my study habits thing that I've been talking to y'all about and what's online. But more than that, believe. Believe first off in God, and believe that He has great things planned for you, and that you are His workmanship created in Christ Jesus for good works. So are you trusting in that? Are you trusting in Him? Or are you trusting in your own strength? We need help. We need help. I need help. You need help. We all need help. And so let's encourage each other. I want to encourage you with that. Like, I'm so proud of you. I know you're working hard. You're doing your best, which is everything that I ask for. And that's all I ask for. You just work hard and do your best for God's glory and for our good. So anyways, I'll stop the video here. 18 minutes and 20 seconds. Hopefully it helps. God bless, guys.